Hello students, welcome to LEELA'S tutorial. So today we will continue the last topic, that is regeneration of plants. In last class we have discussed about the organogenesis, where you are going to produce organs, that is shoot and root system from the explant, followed by production of plantlets. Whereas here today we will discuss about the embryogenesis, where from the explant you are going to develop the embryos, then followed by plantlets. Right? So let us see. In here, the embryo is derived from single somatic cell. Right? The embryo is derived from single somatic cell. Hence, it is referred as somatic embryogenesis. So here, the single somatic cell is having totipotency. That is, the single cell is having ability to transform or divide into a complete plantlet. Right? So that is called as totipotency effect. So because of that totipotency effect of the somatic cell, you are producing an embryo from it and then you are regenerating a complete plant. Okay. So the embryo also consists of both apical and basal meristem. So here you can see the meristem is the rapidly proliferating cells which are present at the tip of the tissues, either the shoot system or the root system. At the tip of the shoot or the root, they are present, right? As a result, they increase the rate of cell division, right? So let us see the process in detail. Select one of the plant. The mature plant, here I have selected the carrot. So the mature carrot is selected, and you are going to excise this explant, right? Excise that root of the carrot plant, and here with the help of scalpel you are going to select a small portion of this cortex. Okay, so here I am going to take a thin section, and from that the central portion I am using as the explant, and then this explant is inoculated under sterile conditions in a plant tissue culture media. Right, so here I have inoculated 3 explants, and then after incubation for 3 weeks, I can see that these cells are getting transformed into embryoid bodies. Right? So these embryoid bodies, further after incubation, they form a clump of cells. Now these clump of cells will initiate this formation of embryo. That means the initiation step is starting from this clump of cells. Once the clump of cells are further incubated with sufficient nutrients, again everything is undergoing in plant tissue culture media only, right? So by taking up the nutrient media or the required nutrients from the media, here these clump of cells get transformed into a globular shaped embryo. So this is the globular stage embryo, which further develops into a heart-shaped embryo, then followed by torpedo-shaped embryo. Torpedo-shaped, where it consists of a long elongated tail. Okay, so at this embryo stage, this is the last embryo stage. Further, it will enter into an cotyledonary stage, where the cotyledons are present. Now from these cotyledons, within 4 weeks, the shoot can be observed. Then it is transferred into the shooting media, followed by rooting media. So here you can see within 4 weeks, a complete plantlet is formed. Now this plantlet will be allowed to grow in, or allowed for acclimatization or hardening process in greenhouse. Then from the greenhouse, they are then planted into the soil. Okay, so like that you can produce a mature plant by simply using the somatic cell. Okay, so here by somatic cell culture you can produce a complete plantlet. Okay, so now it is referred as direct embryogenesis, where the explant is developing into an embryo, and from the embryo you are generating a complete plantlet. So it is an direct embryogenesis. If you want to produce an indirect embryogenesis, if you want to follow indirect embryogenesis, here it will be a long process. See here, the explant, whatever you have inoculated, within 3 weeks you are getting embryoid bodies. right? If you want to go for indirect embryogenesis, what you have to do? Here you are going to use auxin to cytokinin 1:1 ratio, so that without forming embryoid bodies, these explants will develop into an undifferentiated mass of cells or callus. Now, after formation of the callus, again the callus will be induced to form the embryoid bodies or directly the shoot and root system. Okay, so that will be a lengthy process. Okay, no one will go for indirect embryogenesis. They will prefer direct embryogenesis, which is received or which is produced within 3 weeks. Right? If you go for this process, it will be more than 6 weeks or 8 weeks to reach up to this cell clump stage. Right? So, in order to reduce the time, here we go always prefer the direct embryogenesis, where the explant will produce embryoid bodies, then followed by mature embryo, then from the embryo cotyledonary stage, then complete plantlet. Is that clear? So this is very simple process of embryogenesis, but it will take time and you have to maintain complete sterilized condition, and each and every step is a task, right? It is not that easy how we explain. Okay, so in the lab condition, we must be very much careful with the explant, the media composition, internal and external environment, everything must be monitored and maintained. Then only will be success in producing the plantlet. Is that clear? If you have any doubts, put it in comments and subscribe for further videos.